And welcome to the Evidence and Reasons Academy. And today we're going to talk about a paper just briefly, and this is kind of educational. And also, I did a show on zinc fingers earlier on the Examining Evolution channel. So, the topic I'm going to cover is this paper by, let me see if I could show it here. It is the Journal of Psychiatry Investigation. Journal of Psychiatry Investigation. Biological Studies on Alcohol-Induced Neuronal Damage. And this is kind of an old paper, March 2008. And let's just read the abstract, and this will be kind of a short show here. Alcohol is a well-known cytotoxic agent. Cytotoxic. Okay, so since I don't quite know what that means, let me see if I could find another. Hang on. Let me find the, oh boy. Cytotoxic. One moment. So this is actually how we're going to go through and read papers that we don't quite understand. Here it is, cytotoxic, cytotoxic. And cytotoxic. Cytotoxic means toxic to living cells. So maybe what I'll do, just so we have a little bit of a flow, I'll just read through the abstract first, and then we'll go back and try to understand all the terms. So, oh goodness, I have too many windows open. Where did I? Okay. There it is. Apologies. So here we go. Alcohol is a well-known cytotoxic agent which causes various kinds of neuronal damage in spite of thousands of published. Okay, alcohol is a well-known cytotoxic agent. That means something that damages cells, which causes various kinds of neuronal damage. That means nerves. And that would include the nerves in the brain. In spite of thousands of published studies, the true mechanism of alcohol-induced neuronal damage remains unclear. Neurogenesis is the generation of neurons from neural stem cells, NSCs, and occurs predominantly in two regions of the brain, the subventricular zone and the dentate gyrus of the hippocampus, NSCs are the self-renewing, multipotent precursor cells of neurons, astrocytes, and oligodendrocytes in the central nervous system. Recent studies have begun to illuminate the role of neurogenesis in the biological and cellular basis of psychiatric disorders and several clinical symptoms seen in alcoholism, such as depression, cognitive impairment, underlying stress, and brain atrophy, have been linked to impaired neurogenesis. Heavy alcohol consumption decreases neurogenesis in animals, while in vitro studies have shown decreased generation of new neurons after alcohol exposure. These findings suggest that decreased neurogenesis is important in the pathophysiology of alcoholism. Neurogenesis can be divided into 4 stages: proliferation, migration, differentiation, and survival. Our in vitro studies on NSCs shows that alcohol decreased neuronal differentiation at doses lower than those that affected cell survival and suggested that neuron restrictive silencer factor, or repressive element 1 silencing transcription factor, NRSF and REST, could be involved in alcohol-induced inhibition of neuronal differentiation. NRSF and RST, I believe, are zinc finger proteins. In animal model of fetal alcohol effects of behavioral symptoms, behavioral symptoms improved after NSC transplantation. Neurogenesis could be the target for new strategies to treat alcohol-related disorders. So let's go back and try to study this. So I learned the new word cytotoxic. And let's see. Neural stem cells. So let's look at the word stem cells and just study that, just for the sake of completeness. Stem cell. And so this is how to learn to read a biology paper and you're not familiar with the terms. This is how you can learn. This is how I learned a lot of biology. So stem cells offer great promise for medical treatments. Learn about stem cell types, current and possible uses, and state of the practice. You've heard about stem cells in the news, and perhaps you've wondered if they might help you or a loved one with a serious disease. You may wonder what stem cells are, how they're being used to treat disease and injury, and why they're the subject of such vigorous debate. Here are some answers to frequently asked questions about stem cells. Stem cells are the body's raw materials, cells from which all other cells with specialized functions are generated. Under the right conditions in the body or laboratory, stem cells divide to form more cells called daughter cells. These daughter cells become either new stem cells or specialized cells, differentiation with more specific function, such as blood cells, brain cells, heart muscle cells, or bone cells. No other cell in the body has the natural ability to generate new cell types. So let's look at this picture here. And even though I've studied this before, it's kind of good to review because repetition kind of helps your memory. So we have a stem cell here, and from the stem cell, we can make nerve cells, cardiac cells, blood cells, and liver cells. That's an amazing process, by the way. Totally amazing. So I'll leave it at that. So we have something about neuronal stem cells. So I presume at some stage before we get a full-blown nerve cell, we have kind of an intermediate. We have a stem cell that is just made to make a variety of nerve cells. Neural stem cells. So neurogenesis is the generation of neurons from neural stem cells and occurs in predominantly 2 regions of the brain. So going back to here. So this picture of stem cells, I don't know if there's a specific stem cell called a neural stem cell, but you can just imagine we have these stem cells and they're going to differentiate to be all these varieties of nerve cells. Actually, let me take a pause here. I'm going to show you a picture of nerve cell types. There are so many, it's amazing. Let's see if I can bring up the image here and then I'll show you. There are just so many. Here's one with colors, and there are really, I'm going to just show some of the types. This isn't the best. Let me see. This isn't necessarily the best. I'm just Googling here. Trying to find a really nice picture of this. So what I'm doing here, oh, here's a better picture. What I'm doing here right now is just Googling around, and I'm going to show you the different kinds out there. Let's see if Wikipedia has, no, Wikipedia doesn't have a really good one. All right, so now let me expand this. And you can see we have different kinds of nerve cells. So from this parent, let me see here. From this parent nerve cell type, from this parent stem cell, I'm sorry, we're able to generate this variety of nerve cells. So let me see if I could find it. I have too many browsers open. So imagine a stem cell, and it can differentiate to all these kinds. I talked in one of my talks that I'm going to move here to this channel about Purkinje cells. They're prominent in the brain, and there are all sorts of things like the cells in your, I guess, in your spine. There are other cells that help control your muscles and things for sensing and like for being able to see light, etc. So there are a whole variety of cells that are nerve cells. So that's that as far as that goes. I'm just going to kill this window. And now that we've gotten to stem cells, I'm going to kill that window just so I can manage this and go back to here. Neurogenesis is the generation of neurons from neural stem cells and occurs in predominantly two regions of the brain, the subventricular zone and the dendrite gyrus of the hippocampus. Oh boy. So what do we do with that? Oh my. I totally blew it, guys. I didn't realize I could see the diagrams and you couldn't. Let me try to amend that here. Sincere apologies. I'm looking at one screen. I don't always realize you guys aren't seeing it. So I'm going to have to rebuild all this. So let me make sure I got all my ducks in a row. All right. So this is nerve cell types. Hang on. Cell types. And I have a picture here somewhere. It was a really good one. There. And I'm going to just open it in a new link. Now I'll show it to you guys. I apologize. Man, I totally blew that. Wasn't paying attention. So from that stem cell type, so you just think about like when a human being grows out from a single embryo, it's we call it totipotent stem cells. You can make all the other cells in the body from that one cell. And you can also, that would include all these nerve cells. So I was talking about these in one presentation, the Purkinje cells. And there are all sorts of other kinds that can help you sense light or smell, pressure, so you can touch, taste. And so you have to have all these different kinds of nerve cells associated in all the connection, and then cells that help you think, control muscles, etc., etc. The complexity is just staggering. So let me take this down and we'll continue here. We need to find out what the hippocampus. So sometimes if you Google something like this, the subventricular zone, you may not find anything. But what I think you can do is just go to this here, like the hippocampus, and see if you could, and kind of build your way back. So that's exactly what I'm going to do. I'm going to Google for the hippocampus. So, neural types, hippocampus. So probably a good place to go would be Wikipedia, and we'll go right there to the hippocampus. Hippocampus is a major component of the brain in humans and other vertebrates. Humans and other mammals have two hypocampus, one on each side of the brain. The hippocampus is part of the limbic system, and I don't know what that is, and plays important roles in the consolidation of information from short-term memory to long-term memory, and in spatial memory that enables navigation. The hippocampus is located in the allocortex, and I don't know what that is, with neural projections into the neocortex in humans, as well as primates. The hippocampus, as well as medial pallium, is a structure found in all vertebrates. In humans, it contains two main interlocking parts: the hippocampus proper, called the Ammon's horn, and the dentate gyrus. So let's look at this. See if we can click and look. So here's the brain, and there's the hippocampus. I like the fact that they put it in colors. There, that's the hippocampus. The hippocampus. Hypothalamic nuclei, cingulate gyrus, corpus callosum, thalamus, amygdala. This is incredible. If you're going to be if one wants to become a brain surgeon, you'd have to learn all this. That's incredible. So that's the hippocampus. I didn't realize it was so, I didn't know it was, part of it was right so close. I mean, at the edge of what we consider the brain. I learned something. So, all right, so now back to our paper. It mentioned the dentate gyrus. Dentate gyrus. I can't read this diagram. Dentate gyrus. Man, that is a hard diagram to read. Well, let's just take a look and see. The dentate, that's not working. Aggravating, okay. So, let me see, I'm just gonna Google to see if I can get dentate gyrus and hippocampus. Hippocampus, dentate gyrus. See if we can get a nice picture. You know, the thing is you just get these diagrams and you don't really see it in the context of the entire brain. Okay, finally, here's one. All right. If you excuse me, I'm gonna crank up the air conditioner. Hang on. And Schnoil is here. Nice to see you. I'm just trying to learn some brain anatomy here. So let me try to bring up this image. So let me say, greetings, Schnoil. It's late at night over where you are. Hope you get enough rest. So let me go here. And we'll look at some brain anatomy. So this is the front of the brain. And then there's this hippocampus here. And inside is the dentate gyrus. So we have the dentate gyrus. So that's part of the, see, I could see this so much clearer in context. There's the dentate gyrus. So So now let's go back to our paper. Neurogenesis is the generation of neurons from the neural stem cells and occurs predominantly in two regions of the brain. The subventricular zone and the dentate gyrus. So this is actually kind of amazing. I mean, if I'm reading this right, this is pretty amazing. So let me try to get to that illustration again. I think it's this one. So I don't know if this is the case, but the way I read this, and oh, good. Schnoil, did you miss much? No, we're just learning. We're just learning terminology. I'm kind of showing how to read through a peer-reviewed paper. And if you have no background, I'm just showing you how I do it and how difficult it can be, just so you don't get discouraged, don't get intimidated. Because we have the tools of Google, you're able to, you can read a sentence, and if you have to Google each and every term until you get a little hint of what it's talking about, that's what you have to do, and you can get through it. And you can come away feeling you've learned something. And redeem. Yes, we're going to have some more physics here, guys. I need to touch a little bit on biology. And I just kind of got a little irritated with some people, with one individual who's just telling falsehoods about biology regarding my zinc finger presentation. And so this is a little bit of just exploring zinc fingers more in detail so that we can understand it. And also it makes, rather than making it so abstract, we can learn something about the importance of zinc fingers in the functioning of the brain. And then, you know, we can relate it to like everyday experience, why alcohol can give you, excessive drinking can cause lots of brain damage. And we can go into all the symptoms. And I'm going to just have a series on this. And so you can connect what happens to people at kind of like the everyday experience level, and then you connect it to the cellular level. And so I tell you what, when I read this, I mean, I have in our household, you know, I don't really drink very much, but we had collected gallons and gallons of alcohol over the years, and no one would drink it. So people would bring gifts to the house, and we have bottles of alcohol that had been here. I've been with the family for like 50 years, and it's only like recently I began to open and just kind of sniff, and it's like, well, you know, I'll drink a tablespoon or so. But as I started reading this, I got the dickens scared out of me about what alcohol can do to your brain. Turn it into pudding. It's just a disaster. So since you all came, why don't I read all the bad things that can happen to you just to refresh. So it says here, heavy alcohol consumption decreases neurogenesis in animals. Let me see. Okay, so what are the things that happen? Several clinical symptoms seen in alcoholism, such as depression, cognitive impairment, underlying stress, brain atrophy, which is being brain dead, have been linked to impaired neurogenesis. Heavy alcohol consumption decreases neurogenesis in animals, while in vitro studies have shown decreased generation of neurons after alcohol exposure. And so basically it's just killing the brain and doing all sorts of things that are not so pleasant. So this put the fear, you know, this really put the fear of drinking in me. I mean, we're all exposed to all sorts of toxins, and I think like heat, if you're exposed to a little heat, it's not going to kill you. It's actually probably good for you. But if it's too much all at once, it'll incinerate you. And I think that's kind of what happens with alcoholism. If you get just too much of a dose all at once, definitely it can kill you. But then over a protracted period of time, it'll start to just affect your brain and destroy your brain cells, prevent them from regenerating. But they didn't exactly know why, and it's tied to zinc finger proteins. So that's kind of all that you missed. I'm now just trying to go through this and learn all the terms. So the next term I want to study is the subventricular zone. The subventricular zone. I'm sorry, I sometimes forget that you can't see what I'm seeing. So let's look at the subventricular zone. Subventricular zone. Let's look at subventricular zone neurogenesis. And I'm going to look here. See if I could see some pictures of the brain. Hang on, I'm trying to find a really good picture. Well, let's just look at the Wikipedia entry. All right, subventricular zone. This article may be too technical for most readers. Yeah, no kidding. So I believe right here is the subventricular zone based on this photo here. Let's see. The subventricular zone in a rat brain. So this is where all these nerves are generated. So that's a subventricular zone. The subventricular zone refers to a secondary proliferative zone containing neural progenitor cells, which divide to produce neurons in the process of neurogenesis. The primary neural stem cells of the brain and spinal cord, termed radial glial cells, instead resides in the ventricular zone. Okay, we don't have to get that technical, but just suffice to say, a lot of new nerve cells are generated in this subventricular zone. So let's go back to the article we were studying. Alcohol starts to disrupt the subventricular zone and the dentate gyrus of the hippocampus. So if that's, maybe that's about all that I could digest for today on this. I learned maybe 3 terms today: the hippocampus, the subventricular zone, and the dentate gyrus. And that sometimes, guys, sometimes that's just about all you can do when you're learning. You might go to an hour lecture and it's like, oh, I think I came away with 3 more words. But then if it sticks in your head, then that's great. You've learned something, you're more articulate, and that's great. So they tie it, when the brain is exposed to a lot of alcohol, it affects this NRSF and REST. So let's look at this. NRSF. Okay, so it says here, okay, so let me expand this. And I'm getting better at double checking that you all can see what I'm seeing. To clarify how alcohol affects the direction of the NSC, that's the neural stem cells. To clarify how alcohol affects the direction of NSC differentiation, in vitro studies, in vitro meaning in test tube, in vitro studies focusing on neuron restrictive silencing factor or repressive element 1 silencing transcription factor, NRSF, REST, were performed. Okay, guys, don't get blown away with all that, the terminology. You know, sometimes unless you work with the same chemicals over and over and over again, these chemicals just seem like reading phone numbers. I mean, it feels meaningless. So, for example, if I just gave you a ton of numbers, it's just going to be generally, in general, not always, meaningless to you. But let's say it's the phone number of someone you love, it starts to have more meaning. And so just look at these biochemical names and say, okay, right now it's meaningless. But as you start to connect it to how the body functions and how it's important to your own personal well-being and your health, then it starts to make sense. So for right now, these are just names like NRSF and REST. It's like, okay, if these things are disrupted, it breaks my brain, and that's bad. I end up with pudding brains if I expose this to alcohol. And that's how you can make the connection. It's like, yeah, this is bad if you disrupt it. So let's just read this, and we're almost done for this session. I'm trying to keep it short, and then I'm going to have a talk on economics because I'm having fun today. I'm going to post a 10-minute talk on economics that I think is important. And then I'm going to talk about geomagnetism in another. These will be separate streams, guys. So just so you know. So I'm just about, maybe hopefully in less than 10 minutes we'll wrap this up. To clarify how alcohol affects the NSC differentiation in vitro, in vitro studies focusing on neuron, restrictive silencer factor, or repressive element 1 silencing transcription factor. So just so you understand this neurorestrictive silencing factor, see that's the acronym NRSF, and then repressor element 1 silencing transcription factor, that's REST. Okay, so these are just names, but just know they're important. NRSF and REST is a zinc finger transcription factor, which is composed of an N-terminal repressor domain. So I'm going to give a talk. I'm going to upload a talk on zinc finger protein so you can understand how zinc fingers work. Because you could tell some of my favorite classes of protein are topoisomerase, collagens, and zinc fingers. There are 20,000 maybe proteins or protein types that are alternatively spliced in the humans, about 20,000, and they can alternatively splice to 500,000. I focus on only 3 of those. Well, actually, that's not quite right. You can group those proteins into certain classes, like topoisomerases, like collagens. So they're like, I don't know, probably more than 10 kinds of topoisomerases in the human body, probably roughly 30 collagen types, maybe 2, 3, 4, anywhere from like say 200 to 600 zinc finger proteins in the human body. So those are the 3 major classes that I focus on. You could tell those are my favorite, and the zinc finger is really just a broad category of proteins you can learn about. And so I'll have a show on that if the word zinc finger doesn't mean anything to you here. So let's just go on and just finish out this paragraph. NRSF REST is a zinc finger transcription factor, which is composed of an N-terminal repressor domain, a cluster of 8 zinc fingers that functions as a DNA binding domain, a highly basic region or repeat region, and a C-terminal repressor domain with a single zinc finger motif. Oh boy, that's a loadful. Okay, so when you have a protein that's laid out kind of like a sentence, and you have phrases or words in the sentence, they're just describing the major modular components laid out. So the N-terminal, when you're reading English, you read from left to right. You might, roughly speaking, when you're reading the sequence of amino acids, if you could hypothetically read it from left to right, you start with the N-terminal domain, so that's the left side, figuratively speaking, and the C-terminal domain is the end, toward the right side. Or you could say, like, the left side, top of the paragraph to the very end of the paragraph on the right side. You know what I mean, hopefully. So, and they're just describing all the components going from left to right. the major components of the zinc finger protein. And the biochemists would understand, or the people that study zinc fingers would understand the significance of that. So that was interesting to me. So let me just keep reading this. NRSF REST binds to its target, which is by consensus identified as neuron restricted silencer, element repressor, element 1, and NRSE RE1. These are, again, component names. NRSE RE1 was originally discovered in the promoters of the genes that express SCCG10 in the type 2 sodium channel, and later shown to mediate negative regulation in neuronal genes. Through binding to NRSE RE1 and NRSF REST represses multiple neuronal target genes in non-neuronal tissues, also in undifferentiated neural processes of the CNS. CNS is central nervous system. That's kind of, I guess, your brain and spinal cord. Okay. I'm just trying to read this. Through binding to NRSE. Okay, NRSE RE1 is a sequence of DNA. I'm reading this right. NRSF REST binds to its target, which is by consensus identified as neuron restrictive silencer repressor element 1. So I'm gonna go out a little bit on a limb. The zinc finger NRSF/REST connects to DNA, and that section of DNA is called NRSE RE1. NRSE. It's just kind of like a motif or a sentence or a group of words you'd recognize. So we have kind of these dictionaries of DNA sequences, and that's a particular thing you'd find in the quote unquote, figuratively speaking, a dictionary of sequences. It's just like words. And it has variations in spelling. All right, so let's continue. This paragraph's a lot longer than I thought. Its function is to control the proper timing of neuronal gene expression through neurogenesis. To understand the role of negative regulation of neuronal gene expression by NRSF REST, we investigated the effects of alcohol on the DNA binding activity of NRSF REST. These experiments demonstrated alcohol enhanced the NRSF binding activity to NRSE RE1 in a concentration-dependent manner, and these concentrations inhibited neuronal differentiation without affecting cell survival. Let me try to process how this works. So you have DNA that makes proteins, and those proteins are important for how the cell behaves. You can either, there are two ways to make more of the protein. There are ways to make more of the protein or less. So in some cases, it's always making a lot. If you put a repressor there, it's going to just kind of tell it to stop making so much. So sometimes it will just automatically, you know, it would have a tendency to make a lot. You put a repressor there, it kind of blocks the ability to make it. And so when you put lots of alcohol, this zinc finger will actually attach, and it'll attach too frequently, and it's going to shut it down. So just like a recipe, when you're making food, you don't want to make too much of one ingredient or too little. It'll taste bad. So like, you know, you put seasoning on your food, too little, it's tasteless, and too much can be really bad. And we could find other examples of this. You need to get the right proportions. And so alcohol put on, when someone's a heavy drinker, it's causing these zinc fingers to attach to the DNA more frequently than it should. And when it does that, because it's a repressor, it shuts down the expression of these important proteins to make more brain cells. And so you end up having all these effects where your brain just turns to mush. I have I mean, it's kind of disturbing here. Let me show a picture. Let's see if I can show a picture here. And says heavy drinking can cause a lot of brain damage. So this is a normal brain here, and this is a brain that has a lot of brain damage from alcohol. See, it's actually shrinking and it's losing cells. It's just not a pretty sight, guys. Not a pretty sight. It's losing, it's brain damage. It's shrunken. And let's see what WebMD. And so now we actually know the role of zinc fingers in, alcohol attacks the zinc finger, and then the zinc finger causes the binds to the DNA, and it represses the expression of critical proteins, and those critical proteins not being expressed prevents the generation of new brain cells, and there are other things, and you get a brain damage like that. And let's see what it has to say here. Heavy drinking over a long time can shrink brain cells and lead to alcohol-related brain damage. Heavy drinking over a long time can shrink brain cells and lead to alcohol-related brain damage in certain types of dementia. So let's see, Schnoil had a question. Okay. Oh my goodness, what a good question. I'm going to be in trouble here because you're asking questions I actually don't understand. I've never heard of the word silencer. I mean, I've heard it. So, this is a good enough question that I should look it up right now. So why don't we learn together? Okay, I'm not going to BS you and say, oh, I think I know what this means. Okay, so let's look at a gene. Hang on. Just give me a moment here. See, now we can actually learn something. And for you electrical engineers out there who've done control theory, there's a lot of nonlinear regulation feedback control. So if you've done control theory with feedback control, instead of using electricity and say operational amplifiers or whatever, you use chemicals to implement your control circuit. Your feedback, there's a lot of feedback control circuitry here. When you look at some of the gene regulation, it looks like some of my control theory stuff, except it's more complex. So I remember when doing control theory, you had Nyquist root locus and all that, but that was all linear control. Biology has lots of non-linear control, and we don't know how to model it very well. Not only that, you only had single input, single output for a lot of control theory. This one's multi-input, multi-output. It's just mind-boggling. But anyway, let's try to address Schnoil's question here. So, silencer. Oh my goodness! I think you hit it out of the ballpark. That's an American term for baseball. When the batter hits the ball and he scores by hitting it so hard it flies out of the ballpark. That's in baseball. So let me read this. I may have learned something here. Okay, guys, I'm not into genetics, honestly. Okay, so if you're like, well, Sal didn't know that, a little secret. Everyone who studies biology has their specialty. Like, I don't know anything about dinosaurs. I don't care about dinosaurs. right? So there you had the dinosaur people, you had the geneticist. I'm not really that much into the nuts and bolts of genetics. I have done the specific math of population genetics, but that was very, very specialized. But some of this other stuff, you know, so, but since I am a molecular biophysics researcher, I should learn this when given the opportunity. So it says here in genetics, a silencer is a DNA sequence capable of binding transcription regulation factors called repressors. DNA contains genes and provides a template to produce mRNA. That mRNA is translated into proteins. When a repressor protein binds to the silencer region of DNA, RNA polymerase is prevented from transcribing the DNA sequence into RNA. With transcription blocked, the translation of RNA into proteins is impossible. Impossible. It's impossible. Okay, thus silencers prevent genes from being expressed as proteins. Oh my goodness, I am indebted to you, Schnoil. I did not really know that. So the silencer is the sequence where the repressor protein can attach to bind to. Yes, according to everything I just read, I mean, like just now, thank God for Google. I know that's kind of hard to say sometimes, but thank God for Google. Google's very anti-Christian. That's why I say that. This is really cool. So why don't I mean, just so we can appreciate what this is, let me go here. And this NRSERE1. This is going to be helpful because I think when we can visualize this, it starts to become a little more concrete. NRE. Oh, see, silencer delimited transgenesis, NRSERE1 sequences. So let's see if we can find what an NR. Okay, so this is what we call a logo. It's a DNA logo. And so going to your question here, let me take this down and we can expand this. So you have a stretch of DNA, and when you have the very big letters like that, that means those are really important. If you have that letter, it's almost like it has to be there. When you have these things that are just like small, it means different things could be there. It would tend to have a bunch of C's, but it's not as critical as, like, say, the A and the C here. The T is kind of important. This ACC is really important. So you want to see a CA followed by two other DNA nucleotides, but then you have to have the ACC. And then you need to have like a big G and a big G here. So these are like, I'd say these are really important here. And so that's what they call the consensus sequence. That's why I was saying it's kind of like a dictionary, but it's not exactly, it's not exactly spelled the same way, but there's certain features or motifs that are very important. How this is figured out, it's done, it's a pretty brutal process of experimentation. So they'll take this NERST gene, this NRSF REST protein, get it to bind to different portions of the DNA, and then, like in the test tube, and then you hope it stays connected. When they say the word bind, you hope it stays connected. Then you blow away all the other DNA, and all you have is the protein connected to the DNA, and then you release the protein, and then you sequence all the. This is a brutal process. You sequence all the remaining DNA, and you see it has this consensus sequence. And then you do that for all the cells, and then maybe different organisms. It's a brutal process to construct this consensus sequence. So, going back to our genetics, a silencer is a DNA sequence. So this is a silencer right here. So if you saw this sequence in the gene region, you'd probably presume that's a silencer. That's a silencer right there. And the repressor will, let's see, let's see if we just NRSF rest, and see if we can get a diagram. I love diagrams. See if we can get a diagram. Ah, beautiful, beautiful. So here's the repressor, the rest, NRSF zinc finger, and you could see the fingers. I'm pretty sure this represents the fingers of the zinc finger protein. So the fingers there will grab. That's a good analogy. It's going to just like a claw, it's going to grab the silencer region. And what happens is if there's lots of alcohol that someone was binge drinking or whatever, it's going to cause this to grab more frequently. And when it does that, it starts to shut it down. So like I said, like in electrical engineering, you actually build things, we call them resistors, because you want to limit how much electricity is flowing. And so you build this network where you're amplifying, and then you're also putting resistors and amplifiers. You can call it like attenuating or whatever. And you're constantly, and you're building these circuits to be able to do regulation and feedback regulation, feedback control. And so you're like, well, why would you try to shut stuff down? Don't you want to make it? Couldn't you figure a different way to do that? Well, there's a design, it's like an analog computation. There is digital control, digital feedback control. There's also analog feedback control, which was like kind of the, where a lot of the original stuff used to be analog based control. We were able to get a little bit more precision with digital control, but there's still a lot of analog because it's so fast. So if you need high-speed applications, you probably try to find an analog solution if your digital is not fast enough. But anyway, so let's show this diagram again. This is a really great interaction. This is the silencer here. That's on the DNA. So this is the DNA double helix. The silencer is here. The zinc finger rest NRSF is there. And when there's lots of alcohol, it tends to bind more frequently. It has a higher probability of binding and shutting down the cells. And the net result is you get all this brain damage. Let's see if I have the picture of the brain damage. I'm going to kill this. I don't need that anymore. Yeah, you get all this brain damage. That's the net result. So good questions, and I think this went on a little longer than I thought, but I have a couple more diagrams that I wanted, one more basic diagram that I wanted to show. Okay, here is the diagram I wanted to show. This is the CTCF protein that I talked about on Rebecca's Examining Evolution, and I'm gonna, you can refer to that. But this one has 11 zinc finger domains. So you could see here, you see this string, look carefully, it has all these English alphabetical letters. Well, that's the spelling of the protein. This is really beautiful. This is like an incredibly beautiful diagram because you could actually see the spelling of the protein like that, where M is like for methionine and A is for alanine. I don't remember off the top of my head all the names. I think D is like aspartic acid, D valine, or arginine, R is arginine. And you could see the spelling, and each of these here is you could see kind of that protrusion. We'll call that a zinc finger. And you see the zinc ion here. So let me see if I can show it. So you could see wherever the zinc ion connects, that defines a zinc finger in the protein. And you can identify the parts of the protein. You have the individual zinc fingers. This is kind of on the left side, as I said, that's the N-terminal domain, and there could be all sorts of stuff here. And this is the C-terminal domain because it's toward the right side. So we have this region, this part here, the phosphorylation sites. That's a whole nother topic. And then we have the SH3 binding domain. So there are these proteins that have I call the Shirk homology domain. So other proteins can bind here. So this is really cool because you have a protein that connects here, another protein called SH3 that have SH3 binding domains. And then you have the CTCF bind to the DNA, connect to the DNA. And all 11 zinc fingers can play a role. Not always, but many times. And it can bind, I think something, I saw numbers in 80,000 positions. And just watch that video. So why am I pointing this out? I got a pudding-brained response in the comments, and I was really upset. The dude just is clueless, whoever wrote it. He said, well, you don't really, it doesn't really use all these other ones. I'm just like, dude, this diagram totally blows that away. Just quit, just give up, dude. You're out of your league. You don't know what you're talking about. You just spew garbage. That's just the pudding-brain response, and I'm just fed up with it. So I just needed to contest that. But I thought this would be productive, so let me see what Schnoll has to say before I say goodbye, unless this is a really involved question. You would call this whole right region the C-terminal domain? Yes. Okay, so now let me just point something out. This makes sense if you're looking at it from a particular perspective. All right, if you're looking from the other perspective, left would be right and right would be left. But conventionally, when we spell the protein out, like in protein databases, you'd see it listed like in a paragraph way, and it would be read left to right. So that's just a convention. I'm not saying physically it's that way, because left and right is in the eye of the beholder. But the convention is you have this N-terminal domain, and that means there's a certain chemical property here. That would be the amino terminus, I think. Let me get this right. And the N-terminus. N-terminus. Let me get this right, because I'm going to totally... The N-terminus, let's see, Wikipedia. The N-terminus, also known as the amino terminus, is the start of a protein polypeptide, referring to the free amine group. Okay, just think, okay, when you're reading the spelling of a protein in a protein database, and you're reading it left to right, just remember the one on the left is called the N-terminus. It's a chemical thing. So that's how come it got that. The C-terminus is the carboxy terminus or carboxyl terminus. So let me just so I don't botch this up, carboxy terminus, C-terminus. The C-terminus, carboxyl terminus, the carboxy terminus, the C-terminal tail, the C-terminal end, COOH terminus, and that's how come it's called that, because of the chemicals. We don't really need to get down that low. The biochemists just love that stuff, the organic chemists. But going back to our diagram, when you are looking at, okay, I'm going to kill this. That's the silencer. Let's go back to the this one. So you could I don't know if you could see it here at the very end. I don't know if you could see my little pointer here, my mouse with the magnifying glass. Let me see if I can magnify it a little more. See if I can magnify. So the very last letter is an R for arginine. I think D is aspartic acid. I'm going to be embarrassed here. So you have an arginine, and then at the end of that arginine, you have this chemical here, the carboxy group, carboxyl terminus. But you know, just from pure convention, and that's all it is when I'm reading the protein diagram, the left, the start of the paragraph, so to speak, is the N terminus. The end of the paragraph is the C terminus. So I hope that's helpful. So yes, I call the whole right region the C terminal. Interesting. Okay, so how do you know how many actually occupy the C terminal? It tends to be just on a protein specific case by case basis. So it's like, oh, okay, well, is this the C terminus, just that many, or do we go this far? It just depends on the convention that the researchers have adopted, what section they're going to call the C terminus. So like for topoisomerase, I think it's the last 400 amino acids, but the C terminus on another protein could be a smaller or bigger number than 400 amino acids. It's just kind of a conceptual way that we group things together. It's partly also how it functions. So it's very clear this region here has a very specific function with zinc fingers. This region here has what we call post-translational modifications. This is like where you have Christmas ornaments added to the protein to help it. It ends up being like kind of a digital signaling to the other proteins what to do, and it also alters shape and chemical function, etc. It's just amazing. So this is almost, hang on, I just realized you're not seeing what I'm seeing. So this region here is clearly the zinc fingers. This attaches to the DNA to what we call the CTCF binding site. And I'm really sorry about that, Shnoil. I messed up again. So as I was saying, C terminal is kind of what determines what the C terminus is on or the C terminal domain. It's on a per protein basis, and it's just a convention that the researchers have adopted. So we could say this is the zinc finger array here. This region where we have the phosphorylation sites is like kind of working like where we have Christmas ornaments. It's kind of like a digital modification, and you're almost reprogramming the protein to behave differently, and it also signals to other proteins the state of that protein. So there's a computation involved. They're what we call readers, writers, and erasers to do to change the phosphorylation state. So just think of phosphorylation as like a Christmas ornament. Think of phosphorylation. Let me go here. Think of phosphorylation as kind of like a Christmas ornament, and there are all sorts of chemical modifications that you could put on the protein. And these Christmas ornaments, figuratively speaking, will store information that other proteins can judge its state, and it also modifies the behavior of the protein. And this is just amazing. To do this, to change these ornaments, you can put them on or remove them. So it's like readers, writers, and erasers. And so we have a whole suite of proteins that will be just like read heads and write heads, and then readers. It's amazing. It's amazing. Read, write, and erase. And so we have a whole region of the protein that's dedicated to that. And then we have another region of the protein where other proteins can connect to it. And when it says a binding domain, that means when you have a particular spelling here, proteins that recognize that can attach to it. And there are all sorts of proteins that can attach there. And then you have the remainder. So I don't know what the researchers would call the C-terminal domain, but you know, it could be all this, or it could be this and forward, or just this. It depends. It's just the convention of the researchers. So this is great, Shnoil. See, now this is what makes me happy. You are a graduate student in computer science. You don't need 4 years of biology and then graduate school to understand the significance of this as far as kind of like the engineering perspective, to understand the intricacy and just the wonder of this. It just takes a little bit of effort and, kind of going through the tedium of reading through the articles. And that's what I had to do. Just like what I read through there is kind of tedious and boring. But then in the end, you begin to learn something and you're like, wow, this is really cool. This is a way of looking at proteins and the way the body works in a way I didn't see before. And it says in the Psalms, we're fearfully and wonderfully made. So Jesus said, consider the lilies of the valley, how they grow. They neither toil nor spin. Yet I tell you, Solomon in all his glory was not arrayed as one of these. And that was in the passage where Jesus was encouraging people not to be anxious about the future. So his cure for not being anxious about the future is to look at biology. He cited two biological examples. He cited the lilies of the valley and the birds of heaven. And he said, look, you look at the lily of the valley, you have the grass that's here today and gone tomorrow and tossed in the oven. How much more are you of value to your heavenly Father? Because he understands all of this. God does. And so, if anything, studying this biology, looking at the creation, it's a comfort in troubled times. And I'm going to upload a video shortly after I end this stream. And sorry, I was hoping to be just done in 15 minutes, but this is a great discussion. It's going to talk about kind of some troubling economic things. So God has given us the comfort in biology to reassure us he has a great mind. And if he made all this, and even though we're cursed, he has the ability to redeem us. And Jesus is comforting us by saying, look into biology to give you a little bit of hope, because things can look really, really bad. And don't be anxious. So this is one reason I enjoy studying creation science and intelligent design. It's not to debate. It's to find encouragement and inspiration to keep carrying on. If we know, God has left evidence that he's there, and we can be comforted that he's there and that he surely hears us because he understands every molecule and every part of your body and every cell of your body. So in about, say 10 minutes, I'll be releasing that video about China. And I want to thank everyone that has joined us live and who's joining us via recording, the Christian brothers and sisters most especially. So take care and God bless you. And I'll see you in a little bit, maybe later today or sometime soon, to study geomagnetism. But before that, we'll have a video I found on Chinese economics. It's only about 11 minutes. It's going to be very powerful. And oh my, Janet Duttridge, praise God you're here. Hug Sal, I'm running on empty but sending love. All right, let's pray for you, Janet. For the viewers who don't know, Janet is a widow and she's disabled, and Jesus has a real heart for people like Janet. It says in the New Testament, true religion is caring for widows and orphans in their distress. And there's not a lot I could do for Janet physically in terms of fixing all of the problems with her house and healing her body, but we're trusting that this momentary light affliction is building for her an eternal way to glory far beyond all comparison. But that being said, I'm going to say a prayer for you. I'm going to say a prayer for Schnoil, and there's one other woman I want to pray for. Let me do that right now. Heavenly Father, in the name of our Lord and Savior Jesus Christ, I lift up Janet Duttridge. She's running on empty, and yet she's sending love. I thank you for the inspiration she's been to me to keep carrying on. Lord, she has lost a beloved husband, and now she has to carry on by herself without him, the love of her life. And her body is suffering from what all bodies eventually must suffer because of the curse. But I pray you would give her comfort and strength to carry on, to bear her cross. And that's kind of hard to say because I'd rather the cross just be taken from her. And Lord, to the extent you can lessen the burden of that weight in this life, May it be so, just so she's not crushed, that she could have a little more hope. But I thank you that it is building for her an eternal way to glory. And Lord, help with the plumbing issues in her house. Lord, get that plumber to get there quickly and to finish what he needs to, so that that house is alleviated. And Janet has many other concerns, and I just leave those in your hands. I lift up Schnoil and his graduate studies. I thank you that a student of science has taken an interest in my channel. I'm so inspired, and I feel so unworthy of the you know, all the interest in that's been directed to some of the boring material that I throw out on the internet. And so I lift up Schnoil, I lift up his dear mother, and I thank you for the miracles you've worked in that family. I also lift up someone who goes by the handle of Hope. She has she's fighting a divorce settlement and custody battle, and it involves a kid that is that she says is trying to transition from being a natural born girl into this transgendered boy, and it's just causing, it contributed to the custody battle, and she's lost basically ability to contact her daughter because here in Fairfax County, it's not a conservative county, and they ruled against her as if she were evil, and they, you know, they consider it abusive that she's trying to prevent her daughter from transitioning or something like that. It's breaking her heart. She's seeing her daughter being destroyed, and she's being robbed of time with someone she deeply loves more than anything else in the world. And she's having money issues and any number of things. Lord, I lift her up. You've told us we have not because we ask not. And so I will ask for a big miracle on her behalf, that she could have her daughter back and that her daughter would be in good mental state. It's just too young for an impressionable girl to be doing stuff like that. And so I just lift that up to you in the blessed name of our Lord and Savior, Jesus Christ. Amen. So hugs, Janet. God bless you. And I'm going to close out with Rachmaninoff's 18th variation on a theme of Paganini that was featured in the movie The Story of Three Loves. It was a beautiful ballet scene, and also it was music in the movie Somewhere in Time. And so this is for you, Janet. God bless you. Take care. And in short order, I'll be uploading that show, and I'll be in the side chat on Chinese economics. And then after that, sometime thereafter, I hope tonight, I'll get back to geomagnetism, to the riveting, exciting field of geomagnetism. Take care. God bless you. And much love to everyone.